Okay, well, welcome to lesson 10 of your Pilot Prep Science 10 course. We're still in bio, and for this lesson, you guys will get time to take notes. I know in like the first part of bio, I said, don't bother taking notes. But I changed my mind because I realized I go through slides too fast when I don't give people time to take notes. So, firstly, these first few slides are going to be in this presenter mode because I put in transitions that I want you guys to see just for fun. You'll see. Okay. Oopsies. There we go. Our first topic is plant tissues. First off, in plant tissues, we have one of the plant tissues is the shoot system. The shoot system is everything above ground of the plant. So if you look here at this pineapple, basically anything that's also, it's like anything that's not the roots. So everything from this dirt area and up, that is part of the shoot system. The shoot system is involved in gas exchange. It does photosynthesis. It also protects the plant from disease. Because on some plants you can feel it when you touch their leaves, they have like a waxy layer. This is known as a cuticle. This waxy layer protects the plant protects the plant from disease and sometimes certain bugs. And then on the upper right, I have a picture of also a shoot system. Yes. So expanding on gas exchange, in plants, since they perform photosynthesis, gas exchange is photosynthesis. That's what I'm saying here. Because the plant takes in CO2 and then produces O2. There we go, gas exchange. Moving on, root system, so in the previous lesson, I talked about how there were three main tissue types in plants: dermal tissue, shoot system, and root system. That's not really tissue. Okay, but root system, the opposite of shoot system, is everything below ground. Is exactly what it sounds like. It's roots. The role of the root system is to take in water for the plant and minerals, such as salts and ions from the soil. I will move on. Doop. Next up, how did I organize this presentation? The heck? Here is a type of tissue, a type of plant tissue, ground tissue. The ground tissue makes up the majority of the plant. It contains loosely packed cells to allow diffusion, which I mentioned before in the shoot system. It's for gas exchange. It's a great tissue, very helpful, very important tissue. That's what I'm trying to say here. The ground tissue acts in the stem, it acts as strength and support. But in the roots, it functions as a food and water storage. In the leaves, it performs photosynthesis. And there's two parts of the ground tissue in leaves. So again, 10 out of 10, great tissue, multifunctional, does like everything for the plant, almost everything. Important tissue. The ground tissue, so throughout the plant, oh wait, I mentioned it here. It doesn't just perform photosynthesis in the leaves, which I said here. It also does some photosynthesis where it's located in the stem, but most of the time, like the majority of photosynthesis is performed in the leaves by the ground tissue. Okay, now this is where I exit out of it and start editing because I did not finish making the lesson. So as I mentioned, there are two types of ground tissue in the leaves. The first type is the palisade tissue, where it is located right below the epidermis, or right below, like, so if you look here on the right, it's like a leaf. This is the cells, the inside of the leaf. This weird clear part, that's the cuticle, the waxy layer that protects the plant from the disease. And then this thin part is the epidermis, or the skin of the plant. And right below that, we have the palisade spongy tissue layer. This is for photosynthesis. They're neatly packed and rectangularly shaped. Rectangular box shaped. However you want to word it. And then the second type 2, oopsies, is the spongy mesophyll tissue. I cannot spell for the life of me. Okay. The spongy mesophyll tissue, they are loosely packed and located below the palisade tissue. So if you look back to the diagram here, the palisade tissue is right here, right below the epidermis, tightly packed, rectangular shaped. And then below that, we have the spongy mesophyll tissue. It's spaced out and it is located below the palisade tissue. They're also kind of irregularly shaped. They're loosely packed to allow for gas diffusion. So if you look below the spongy mesophyll tissue into the lower epidermis, you can see that there's like a hole here. This hole is what allows for gas diffusion into and out of the plant leaves. Loosely packed for gas diffusion. Next, third type of tissue, vascular tissue. The vascular tissue is like the plant's circulatory system. And if you don't remember what the circulatory system is, it's just the system that transports blood in your body. So it's like the blood of the plants, but not really. It's a horrible metaphor. First off, you guys probably learned about this since elementary school. I remember learning this in elementary school, the xylem and the phloem part of the plant. First off, xylem transports water. And then the phloem transports food. Easy. Well, not really food. It transports sugars, which you can consider as food for the plant. And then you can remember which one transports which in the same way that I made you guys remember phagocytosis and pinocytosis, where phagocytosis starts with like a f sound, pH. So you remember it to transport food. Here we have phloem, you think phloem, like food. So you can remember that phloem transports food, and therefore xylem being the only other tube, it transports water. And then more on the xylem. The xylem can only transport in one direction. Oops. Whereas the phloem can transport in any direction. So I can use my annotating drawing stuff. Woo. So here we have the xylem, and then it transports water up the plant. Xylem. And then we have the phloem, and it can transfer stuff up or down. So the reason the phloem can transfer its stuff up and down a plant is it's like season changes. In the summer, when it's growing, it needs to transport food to the upper parts of the plant for it to bud flowers, to make fruits, etc. But in the winter, when the plant's dying, it wants to transport all of its food into the roots to kind of store it for next year, like hibernation. You can think of it that way. Whereas water just, you don't need to store water. There's just going to be water. But if there's no water, the plant dies in the end. I shall erase. Next up, we have meristems. This is an area of growth on the plant. It's just the area on the plant that grows. I reworded the same thing. I said the same thing twice. Yeah, by mitosis. So there's more than one meristem on the plant. You can find them on the tip of the plant or in the middle of the plant, or even on the roots. So as you can see, the meristems on the tip of the plant allows it to grow taller. The meristems at the roots makes the roots grow longer. And then there's also meristems in the middle of the plant. So if we look at this, can you see this image on the right here? I will assume you can. When you like shine a light on a plant, that's a rhetorical question, don't answer it. When you shine a light on a plant and then you change the direction of it, the plant bends towards the light. So if you look at the image here, this plant doesn't grow. It doesn't grow any taller when it bends over, but it changes shape. With the area where it bends is known, like its peak bending is the meristem. Because, bro, how do I explain this? Oh, that's all you need to know. You just need to know that meristems are places where the plant grows. Cool. And then here I attached an image of mitosis in case you forgot what mitosis was, which is when one cell just splits into two. And then, next topic. Meristems, chloroplasts. So if you remember, I mentioned that chloroplasts only grow in plant cells. You won't find them in animal cells since they perform photosynthesis, and we don't perform photosynthesis, unfortunately. We should be able to. I wish we could. Be nice. Now, the chloroplast contains a green pigment known as chlorophyll. Oops, I can't spell. Known as chlorophyll. And that's what makes plants green. That's why plants are green. Wow. Majority of chloroplasts, they're found in the tissues and leaves of the plant, sometimes in stems, but usually just the tissues and leaves. Another way to say it is that when looking at plant leaf cells versus plant stem cells, in the cells that are in the leaf of the plant, there's going to be a lot of chloroplasts, versus the cells of the stem, there's going to be much less. They both will still have chloroplasts, just one will have more than the other since it's more specialized to perform photosynthesis, which is the role of the chloroplast. It performs photosynthesis. The chloroplast uses light energy, so sunlight, usually sunlight, artificial light works too, to convert chemical energy, so like chemicals, molecules. Oh no, it just It converts light energy into chemical energy. What am I doing? Converts light energy. Yeah, it converts light energy. Chemical. This isn't 100% correct, but you're in Science 10. You don't need to know the exact process yet. You will learn it in Bio 20. It takes a while to understand. Honestly, it's a long process. Like photosynthesis is genuinely a long process once you learn the in-depth parts of it. So the light energy, it converts into chemical energy, which is glucose, the chemical energy it makes. And then that's where we store the energy. I'll write stored as glucose. Stored as glucose. We pull out a photo of a chloroplast. And then I am just going to, let's take this photo. It's kind of ugly, but I will take this photo and then straight out copyright it onto my lesson. This is now my photo for chloroplast. This is not what I was looking for. You know what, I'll draw my own. My artistry is a 10 out of 10. Let's take a look. Okay, here we have the chloroplast, and the chloroplast has a double membrane. So that means it has like 2 skins. Just think of it that way. It has 2 layers of skin. And then inside the chloroplast, oh, well, we're getting into a lot of stuff here. There is DNA, which makes the chloroplast a very special organelle, because no other organelle, nucleus being different, has kind of their own nucleus. There we go, that's what I'm trying to say here. This basically, the chloroplast has its own nucleus. And then other organelles don't have a nucleus, except for the nucleus, which is just a nucleus. The nucleus itself is a nucleus, and that doesn't count. But the other organelles, like the Golgi apparatus, the endoplasmic reticulum, they do not have their own nucleus. Whereas the chloroplast, and then also the mitochondria, would have their own mini nucleus. This is referred to as circular DNA or prokaryotic DNA. I'm not going to try writing it. It's too much work. And then because there's prokaryotic or circular DNA in chloroplasts and mitochondria, and they're like really powerful organelles, there is a theory that speculates which chloroplasts and mitochondria, they once, a long time ago, they weren't organelles. They used to be their own cell. And that's why they have their own nucleus. But then at some point in time, stuff evolved, and then they became a part of a cell and became an organelle instead of being their own cell. Okay, let's keep talking about the insides of the chloroplast. We have the prokaryotic DNA, the double membrane, and then we have little stacks. Each of these little circles are called grana, and then more than one, like a stack of them, is known as granum. And then the skin of these things are referred to as thylakoid membranes. I need to write these out, actually. Let me just type these out. I'm gonna write it down here. So we have the double membrane, prokaryotic / circular DNA. I cannot spell. / circular DNA. We have the grana / granum, depending on plural or singular, where if at some point in time you have to label a chloroplast, you won't be docked marks if you can't forget the singular or plural. Let's see, grana, and then I'm going to type this out. I'm going to explain it in a sec. So the thylakoid membrane. Okay, so the skin of this stroma here, no, not the stroma, the skin of the grana is referred to as the thylakoid membrane. And then we have the stroma, which is like the cytoplasm of the chloroplast. Wow. And then photosynthesis just takes place within the chloroplast as a whole. Some part of the photosynthesis process occurs in the stroma, which is like this empty space, and part of it occurs in the grana. But you don't need to know that. That's for bio-twinning. I shall move on to the mouse. And then now we talk about photosynthesis. So here's the formula: 6CO2 plus 6H2O. And yes, you will, oops, I can't spell. You will have to memorize this like formula because you're going to use it a lot continuously throughout biology and sometimes even in chemistry 20. Not a lot though. So 6CO2 plus energy, which is light energy. You have to draw an arrow because for some reason in science you don't use equal signs, you use arrows. Just the arrow just means equals. Equals, oops. C6H12O6, which is glucose. And oxygen. It makes oxygen, 6O2. Why is that 6O2? So the stuff on the left here, the CO2 and the H2O, they have not a lot of energy. They are referred to as low energy reactants. So these two things. But then they take in the energy of the light, and then it makes products with the light's energy to create high energy products for use. So let me just erase this horrible chloroplast drawing. And then I'm gonna keep drawing, more drawing. So if you remember on the picture of like the spongy tissue or something, ground tissue, there's the ground tissue, there's like the epidermis, and then there's this, the boxy tissue, it's like the palisade ground tissue. And then we have the spongy mesophyll tissue that looks like this. And then we have the bottom one, the bottom epidermis. And then on the bottom epidermis drawing, there was a hole in one. There was a hole in it. So in the plant leaf cells, there's actually a lot of holes in their bottom epidermis. I'm going to draw more holes in here. These holes are known as stoma. So I'm going to write that down. Stoma. See, yes, they're a stoma. And then this is where how your, again, gas diffusion takes place. Your CO2 enters through these tiny holes on the bottom of your epidermis, and then the process just, whoa. There we go. CO2 enters in, grab this, whoosh, and now your photosynthesis occurs, and then your oxygen comes out through that same hole. And now we have oxygen in the atmosphere. That's what happens. More holes. Let's draw more holes. Okay, and now erase my bad drawing. Next. We now talk about gas exchange. Oh, I just talked about it. Okay, occurs by diffusion. through the stoma. Or you can say stomata. I think stomata is the singular and then stoma is the plural. So you can use either word. And into the air spaces in the plant. Just in the spongy mesothelial area. Yeah, genuinely cannot spell. But, so back to my horrible drawing that I should have kept instead of erasing. Here we have your epidermis, we have your palisade cells, yes, palisade tissue that looks like this, and then we have your spongy mesophyll that kind of looks like that, but not really. And then we have your bottom layer, and then there's holes. Your CO2 comes in here, and it hangs out in this, like, the empty space between the spongy mesophyll cells. And then it goes in here, it kind of does, and then does this photosynthesis. Wow. Okay. Switch back to a mouse. Oh, next slide. So, stoma, or stomata, or plural. And plural. Here, let's see. They are regulated by guard cells. So the stoma is not just a hole. It has guard cells on it. That's what I'm saying here. Greater efficient intake. Greater efficiency in intake of gases. Efficiency in intake of gases. That's what the stoma does. It allows for greater efficiency of gases. See what else it does. Performs the rapid diffusion of CO2 into the palisade tissue cells. So after the CO2 comes into your spongy mesophyll empty space, it goes into the palisade tissue cells for photosynthesis. Now the guard cell on the stoma, they can close and open. So they can close the stoma and open up the stoma to allow for the diffusion, or I guess stop the diffusion. So we should talk more about the guard cells because we need to know more about the guard cells. Okay, so here we have the hole. Let's say we're looking at the leaf from the bottom, and this is the hole in the leaf. And then around it, we actually have this two sausage-looking things. Okay, those are the guard cells. When they're open, it looks like this, and then when they're closed, the guard cells just look like that. It closes up the hole. The thing about this is, how do I explain it? You guys will learn more about, and I think in a later, why is it not in my notes? Do I have to just explain this from the top of my head? Please tell me I don't need to explain this off the top of my head. No, I have to explain it from what I remember of my science course on like a year ago. Okay. The guard cell opens when it's like swollen, and then when it lacks water, it becomes flaccid and it falls limp, closing the hole. So if you've ever learned about survival, like survival course, where if you lack water, you should put leaves in a plastic bag and leave it there for a day, and then by the end of the day, you look in the bag, there's gonna be a lot of water. That's because water leaves the leaves. That didn't go well. Some water leaves the leaves through the stoma. So when the plant has a lot of water, the guard cells, they swell open and some of this water can come out. But when the plant cell is lacking water and is dehydrated, these stoma, like the guard cells, they fall limp because of lack of water, and it prevents any more water from leaving the leaves of the plant. Now, where does the water in the plant come from, besides from coming from the roots up to the leaves? It's also because, wait, give me a second as I Google how cellular respiration is performed, because I forget. Oh, this drawing is still there. Formula. This is just straight out copyright. Ah, yes, that's what I was looking for. Okay, so cellular respiration creates water. It's the opposite process of photosynthesis, and your plant cells also perform cellular respiration, as do human cells. This is when energy in your body is consumed, right? Yes. The intake of oxygen. This is what you use oxygen for. You intake oxygen, and the cellular respiration releases CO2 and water. The plants perform cellular respiration, and water from it goes through your plant leaves. They leave through the stoma. Moving on. Next, we have lenticles. Well, easy. Apparently, I spelled it wrong. Lenticels. All right. My teacher spelled it wrong. Yeah. So if you've ever looked at a tree, and in some trees you will see this. Let me erase my horrible palisade tissue drawing. So I'm gonna assume you guys have seen trees like this at some point in your lifetime. I know I have. And these cut scar-like things, they're called lenticels. Lenticles, lenticels. And you're gonna have to Google how it's pronounced because I do not remember. I'm gonna say lenticles. I'm sorry if I'm wrong. So here we have the usually, yeah, you get what I'm saying here. Lenticels. Cuts, they're like scars in the tree. They look like cuts. Plant. They are just openings on tree stems / bark. And their role is to the role of these things is to provide a pathway for gas exchange. And transpiration, which is water loss, which I mentioned before with cellular respiration and how water leaves through the stoma in leaves of plants. Transpiration is the water loss in plants. Humans do not perform transpiration because we don't, wait, do we? Because like we sweat. So that's like losing water through our skin. I think transpiration is just a plant thing. No. When we transpirate, sweating counts as transpiration, I'm pretty sure. So yeah, transpiration is just the loss of water kind of like through your skin. Okay. You now have a 10-minute break. Come back at like 4:15. You got 10 minutes to chill. See ya. Okay. So again, lenticels, lenticles, lenticels. They are essentially giant stomata. Okay. Now we have a checkpoint. So again, Angela being the only person here, if you can choose to answer these questions whether in chat or by unmuting. First off, shoot system versus root system. Just tell me the difference. You don't need to fully explain them. You can explain to me what the shoot system is, and then just say that root system is the opposite of it.
Okay. So the shoot system is like everything that's above the ground.
Yeah.
The root system is everything that's below it. And the shoot system is involved in gas exchange, while the root system is involved in like the uptake of water and minerals from the soil.
Yes.
And the shoot system protects plants from disease by like secreting the waxy layer known as the cuticle.
Yes, that was beautiful. Okay, that was amazing. That was more than I expected, much better than I expected, and I like that. All right, moving on. There are two types of ground tissue in plants. If you recall, there's the palisade tissue and then the spongy mesophil. Choose one of the above, so the palisade or spongy, and explain what it does. Like, kind of just talk about it.
Okay, so the palisade tissue, it is below the epidermis.
Mm-hmm.
Its shape is neatly packed and about like rectangular shape.
Yep.
It is used for photosynthesis.
Mm-hmm. Now, last part of the checkpoint. Name a role of the stoma. It does a few things. Name one thing it does. One of the things. Or if you want to show off, you can name as much as you know.
The stoma helps like it helps the diffusion of CO2 into the palisade tissue.
Mm-hmm.
And it has a greater efficiency in the intake of gases.
Mm-hmm.
It's also regulated by guard cells.
Yes, beautiful. Okay. I'm gonna move on. Now, our next topic-ish, the next thing we're gonna learn about is the mitochondria. Let's see, it performs cellular respiration. What else does it do? Oh yeah, I need to show you guys the formula for it. You don't need to remember the cellular, you should remember the cellular respiration formula, but you're not gonna use it as much as the formula for photosynthesis. C6H12, where is the formula? There you go, O6 plus. By the way, the numbers should be in subscript, but it's too much work to turn it into subscript every single time. So I'm gonna type it out like this. Plus 602. I'm taking too long to write this formula. This should be an arrow. I can't be bothered to draw an arrow. I'm just gonna write it in equal sign, but in your notes or on your paper, or when you take tests, it should be an arrow. Why do I keep doing that? CO2 plus 6H2O and ATP, which is energy for your body to use. So I'm just gonna write the photosynthesis formula down here for a reference, so you can kind of compare the two. It's honestly the same. 6CO2 plus 6H2. Oopsies. 6H2O, and then arrow to make C6H. Oh wait. It also uses energy, light energy. To make C6H12O6 and like oxygen. Yes. So you can see here the whole purpose of photosynthesis is just to create a kind of a stored energy in this glucose. And once your body processes the glucose, you can turn it into energy, more energy. So again, the whole purpose is to convert light energy into chemical energy, then that which can be used by cells both in human bodies and animal bodies and in plant cells. Okay, keep going. Mitochondria. Now we draw it, like how I try to draw the chloroplast. I'm going to draw the mitochondria and hopefully do a decent job. Okay, first, just like the chloroplast, we'll draw an oval with a double membrane. Beautiful double membrane here. Let's see. The insides, it kind of looks weird. I'm gonna Google a photo. My drawing ability is really sad. More copyright. You see how the inside is like squiggly. This was a horrible version of the squiggly insides. I'm gonna redraw my squiggly insides. Squiggly insides. There we go. That's my drawing of the mitochondria. Looks about the same, obviously. And then, again, it has its own circular DNA or prokaryotic DNA, which makes it special. Now that you have seen what it should look like, let's go back to my lesson slide. And then I'm gonna write here, I'm gonna write it down as a double membrane. Oopsies, membrane. It has folds, which is these wrinkly folds things, which are called cristae. And I'm going to say it again. It is for surface area, and biology and everything, every other living thing in biology loves surface area. The purpose of these folds is to increase the surface area. We have the matrix, which is kind of just this whole thing in general. All the folds in general are referred to as a matrix because it looks like a matrix, kind of, not really. And then we have our circle, the circular DNA. That's it. I think that's it. Yeah. My drawing covers up the... See if I can move my drawing. No, I cannot move my drawing. Oh, I can. There, now you can see my notes while looking at my drawing. No, the circular DNA. Please, yes. There we go. Nope. Can I move the whole thing at once? Yes, I can. That would have made everything easier. Okay, well now I know. I can change the color. I can, oh, no. Okay. Anyway, I'm gonna assume you're done your notes, so I will move on. Whoosh. Oopsies. Go. Now, I'm back to the dermal tissue, which is the epidermis. And as you mentioned, it secretes a waxy layer referred to as, I think, the cuticle. Yeah. Located on the top and the bottom of the leaf. You honestly don't need to write this down because it's like, you kind of know it. Since it's the skin, it's just going to be on the outside, which is the top and bottom. Leaf. Wait, no, just on top and bottom in general. And it allows gases in and out by having holes in it. And the epidermis is where the stomata are located, which includes the guard cells. where the stomata, cess, and the guard cells are located. And again, guard cells control the opening and closing of the stoma. We can move on from this. Simple information. Now, we talk more about the guard cells. Okay, for me, the information about the guard cells when I was learning it was quite a bit of information to absorb. I'm going to type this out and then attempt to explain while I type. Light stimulates the guard cells well to gain potassium ions, or K+, and the plus sign should be in superscript. Format, text, potassium ions. This causes the guard cells to become hypertonic. So if you remember what that means, it means, wait, let me draw it. I want to draw with my new color. Here we have your guard cells. It is flaccid right now. It is not open. That was horrible. But pretend this is a great drawing of two guard cells being closed. And then light goes whoosh, whoosh. And now K+ goes in, which is potassium ions from just your environment. For whatever reason, there's potassium ions in our environment. And it goes in. This causes the guard cells. I'm going to redraw my guard cells, but better this time. Oh, it is better. That is beautiful. I need to stop saying that word. So let's say these are the potassium ions, the dots inside the stoma. Why aren't my dots forming? Ah, okay, that is a dot. Those are dots. And outside in your environment, there's only a few dots. And that makes the guard cells hypertonic because they have more solute in them than the environment does. What happens when, do you recall what happens when something is hypertonic? Here's a hint, the water goes somewhere. Water is important. And a further hint, water either goes in, does water go in or out in a hypertonic situation? You can answer in chat or by unmuting, or just say, I don't know. That's honestly fine. Okay, I will continue. Because the cells are hypertonic and there's more solution in, there's more solute, so the K+ inside the guard cells, there's like a lower water concentration, which means there's a higher water concentration out here in the environment. And then since stuff goes from high concentration to lower concentration, osmosis occurs. And now this water just goes, all the water goes into the flaccid guard cells, which are currently limp, making the stoma closed. And then the water going in causes the guard cells to swell, and they swell open, and they create their kidney shape. And now there's a hole. That is the stoma. And those are the guard cells. And here's the K+ ions. I need to draw arrows. Sorry, whoosh. And then this goes to this. Water goes in. to neutralize the hypertonic sound. And then because the water goes in, it's under turgor pressure. And again, let cells swell open. Through. We are covering. Come on. Yes. No. Yes. Oh, there we go. Put my horrible drawing down there so you can see the notes. So plants in dry climates like deserts or just areas that don't rain a lot in general, they have less stoma. This is an adaptation to prevent too much water from leaving the plant. I can now erase my drawing. Let us move on. Next up, we talk about the phloem. So again, food transport, sugar transport. Transport in any direction, so it can transport up and down. And the movement of the sugar or sap. So if you break open the phloem, the sugar is kind of like in a sap form, syrupy. Is described by the pressure flow theory. And you don't need to know what the pressure flow theory is. You just need to know that's described by the pressure flow theory. And it is composed of, the phloem is composed of sieve tube cells. And I'll copy paste an image in a sec to show you what it looks like. No, I can draw one. Sieve tube cells and companion cells. Never mind, I cannot draw companion cells. But I will attempt to draw anyway, and then find a photo so you can have extra reference. So here you have your phloem, which is like the tube. And then in your phloem you have sieves, basically, that's it. Well, that's not it. I'm gonna keep going, but that's what the sieve tube cells are. And your sieve tube cells do not have a nucleus. So another way to say it is they don't have a brain. To make up for that, on your phloem you have companion cells, which are on the sides of the phloem. So in here you have your sugars that kind of just go around through the sieves. And then on the outside you have your sieve tube cells, your companion cells. And they have a nucleus. The companion cells, the nuclei of the companion cells, they kind of also work as a nuclei for the sieve tube cells. Yeah. Let me draw one here. Yes. I will show the image. I'll copyright an image in a second. Okay, time to copyright an image. Phloem seed tube cells. There we go. Where is it? Here you go. That is a good drawing. That is a good image. Here's one more drawing. So you see here, this is your seed tube cell, and then it has seed-like holes, which connects the passage to other seed tube cells. And then on the side of your seed tube cells, you're going to have these companion cells, and these companion cells will have a nucleus. Yeah. And let's come back to the notes. Go back to the notes. Oopsies. Next, roots. They use ATP. All your cells use ATP. But what do your roots use ATP for? They use your ATP for the intake of ions or minerals. And if you recall, ions and minerals, these are wait, what? No, ATP, that is energy. So I'm gonna draw your roots. That's your tree, and then it's gonna be a tree. Oh, okay. And then it's gonna have apples. They don't look like apples. That makes me sad. Let's draw a big apple. And then we're gonna have Newton here because your previous unit was physics, so Newton is relevant. Anyway, roots. They use energy, ATP, and the best way to kind of describe it without going into too much unnecessary detail is uses energy to force the floating ions and minerals around it. ATP kind of forces the surrounding ions and minerals to go into the roots. Next. So all you need to know is that roots use ATP for the intake of ions or minerals. Now we get to talk more about the ground tissue. So again, the spongy mesophyll tissue, or you can just call it mesophyll tissue. What is this? What is my planned lesson? Oh, you heard nothing. What is this? What have I written down here? What is between upper and lower? No, it's just a repeat of what I wrote earlier. We're gonna skip this. It's a repeat. Moving on, vascular tissue. What is vascular tissue? They are the plant veins. So the xylem and phloem. That one. Oopsies. So quick review, xylem transports water in one direction. Phloem, food, any direction. And together, the xylem and phloem create a vascular bundle. So first, let me erase this horrible drawing. And then we're gonna pretend we're looking at, let's say this is a plant stem, and we're looking at it from above. I've cut a plant in half, and then we're looking at it from above, the stem. Here we have the xylem and the phloem. So one of them is bigger than the other, and I don't remember if it's the phloem that's bigger or the xylem that's bigger. And I'm pretty sure it's the phloem. And usually when you look at a plant, it's pretty obvious. So like it's gonna probably look like this, let's say. And these are your vascular tissues. I'm gonna check in a second if I'm wrong, but we're gonna say so these all the large ones, they're the phloem, and all the small ones are the xylem. And then this whole, like all these circles as a whole is referred to as a vascular bundle. So I'm gonna look, let's see if I can find an image of a vascular bundle. There we go. Let's erase this. Oh, here we have a real drawing, like a real one. Oh, here we go. It kind of, not gonna lie, kind of looks disgusting. Well, actually, that's up to you. Maybe you like looking at photos of vascular bundles, but I personally do not. It looks kind of disturbing to me. So you can see here we have our phloem, the larger holes. And then, wait, I can't tell what's happening here. And then these smaller holes, I'm gonna say, are xylem. This is not a great diagram. Let's see if I can get a better one. Where are the ones that show xylem? Okay, you know what? I'm such a great instructor. Are phloem or the xylem bigger? I can't tell which one. Or is the xylem bigger? This is a learning moment for both of us. Phloem condenses are smaller and occupy a slightly larger fraction, 10 times more xylem than phloem. What? Okay, xylem is larger because it transports minerals and water. Well, xylem is the larger one. Looks like I was wrong. Now, the arrangement of xylem and phloem in the vascular bundle differs between plants. So if you look at this one, we have this, like, we have kind of 4 giant stuff. Wait, no, that's not what I'm looking for. Here we go. This is a better image. Primary phloem. Yeah. So we have phloem that's small, and then we have the xylem that's kind of spread around. Sometimes they have, so depending on the plant, this is an example of a vascular bundle in a plant. We have the xylem here, and we have the phloem. Their arrangements differ. So in some plants, they might look like this, where it's neatly organized. The pink area is the xylem, the blue area is the phloem. Yeah. Xylem is smaller. Xylem is bigger. Sorry, xylem is bigger. Why is it not coming up? Xylem is bigger. Okay. Next. Jokes, I'm not done with this. Xylem is bigger. Why do plants need to put water? No, I'm actually done with this. Next topic. H2O transport in plants. So there's more to it than just xylem. All substances transported in the plant must be dissolved in water. So if you remember, the phloem transports sugar in a sap-like consistency. That's because the sugar is dissolved in water. are the factors that affect water transport in the plant? Take a look is root hair. So what that means is roots have micro roots. And you can probably see it. You have your root, and then the root has more roots, and that root has more roots, and it has more roots, and that's root hair. These are, and then there's micro roots, to the point that there's tiny roots that cannot be seen with the naked eye. Okay, water transport, root hair, transpiration, and pressure. Factors affecting water transport in water. So by this I mean pressure inside the plant, and you will learn more about this in pretty soon. Maybe on the next slide. Not on the next slide, but soon. Now, we're going to talk more about root hair. What is its purpose? Guess what? To increase the surface area. Root hair, transpiration, water. Okay. And then we talk more about transpiration. Transpiration. The definition for kind of definition for transpiration in plants: evaporation of water through stomata and guard cells and lenticels and lenticels and lenticels and lenticles, creating tension or transpiration, also known as transpiration flow. So what this means, or what happens, is as water evaporates from the plant, or as water leaves the plant through the stoma, evaporating, it pulls on nearby water molecules. Since water is hydrophilic, so that means it's attracted to water. Water is attracted to itself. Like, think of magnets. They have attractions. They attract each other. Water attracts itself. So as water evaporates, as water leaves the plant, it pulls on nearby water while it leaves. Let me draw. Yeah, here we have your plant, okay? And then here we have your leaf. And your water is evaporating. And there's water like here. There's water in here. So how does water defy gravity by going into the leaves of your plant? By having water to evaporate. So again, water here, as it evaporates, this chain of water essentially is linked and connected together, and it's kind of this one water evaporating pulls along this entire water chain, causing the water to move up and to continue evaporating in the transpiration process. So think of the water as like along the chain. Now, this is the attraction of water to itself is referred to as cohesion. Water also sticks to other things, like to non-water, to non-water. Anything non-water is referred to as adhesion. And this adhesion and cohesion works together to make water defy gravity. Gravity in plants. So water can move up the plants. And then, so an example of adhesion or how adhesion works is, let's take a look. If you've ever looked in a thin tube and then you filled it with water, and actually when the water is filled up, it actually doesn't look like this. Let's say this is the water level, okay? Here's your bottle, okay? And I filled it with water. It doesn't look like this. It doesn't look like a straight line. What it actually looks like is like this. There's like a this curve is referred to as a meniscus. Let me see if I can find you an image of a meniscus. Water. Oopsies, let's have an eraser. Rid of my horrible drawings. Here's a better image of meniscus. So because water sticks to other things, in this case, the water is sticking to the side of this glass tubing, causing it to have a curved shape. And that is referred to as a meniscus. This is the adhesion. And then here we can see a good example of cohesion. So even though the water is outside of the glass tubing, it's still sticking together and not kind of overflowing. Oopsies. So coming back here, this adhesion, so the attraction of water to kind of the sides of the xylem, and the cohesion, so the water being pulled up via the evaporation process, it works together to defy gravity. Now, when there's a higher temperature, there is more evaporation. So more water being lost. More evaporation or more transpiration. Remaining water that is not transpirated is used to process sugars / perform photosynthesis. So if you remember the photosynthesis formula, it uses water. Okay, so I would recommend knowing the definition of adhesion and cohesion, where one is the attraction of water to itself, and then the other is attraction of water to non-water stuff, certain non-water stuff. Water is not attracted to everything that is non-water. Now we talk about something referred to as tropism. Oh, I might be wrapping up. Yeah, we're actually nearing the end of the bio unit. We're like really close. So tropism is the controls when the control system of the plants work in response to external stimuli. This will make more sense really soon. External to ensure plant survival. Let me erase this drawing. And then we're going to learn about two types of tropism. Phototropism, and gravitropism. There might be more types, maybe not, but we don't need to know those. First up, phototropism. Phototropism. Plants what? Bend. Oh, plants bend towards light. Plants bend towards light for photosynthesis. And that is how we're going to conclude today's lesson. There's more to phototropism. Phototropism will continue on in the next week's class. And tropism is the last sort of mini topic. So once you're finished learning about phototropism and gravitropism, that is the end of the bio unit for Science 10. You are going to be done. So next class will be, we're going to start on global systems. Because I know I said it was going to be optional and I might not cover it, but we're going to cover it. And Tony will be teaching global systems. Or he should be. What else is there? Oh, wait, I need to pause my recording. Goodbye to the people who are watching recording. You